Angela, can you hear me now?
Yes, I can hear you.
Okay, perfect. So my apologies. Welcome to our program on what the immune system does when I have breast cancer. We are very fortunate to have Dr. Zernicki with us today to present this webinar. A couple of features for you is that we have the chat box open. So if you have any questions as we're going through the webinar, just please place them in that chat box, and we will certainly get to those questions and answers at the end of our program. At this moment, I'd like to take a moment just to read a message from our compliance department. The content of this program is not intended to be medical advice, and the viewers should consult their physician should they have any questions. Viewers should not only rely on information contained in this webinar for urgent medical needs. Additionally, if you think you may have a medical emergency, please call your physician or go to the nearest emergency department or call 911 immediately. Never disregard professional medical advice or delay seeking care because of information contained in this webinar. I'd like to take the moment just to introduce Dr. Brian Zernicki to you. Dr. Brian Zernicki is our department chair and senior member in the Moffitt Cancer Department of Breast Oncology. His research goal is the development of vaccines for the prevention of breast and other solid cancer or solid tumor cancers. Toward that end, he is working on identifying molecular targets in early breast cancer that can be used to prevent invasion and metastasis. Dr. Zernicki has more than 100 publications and is recognized nationally for his contribution to the development of sentinel lymph node mapping, a procedure for determining the spread of cancer into the lymph nodes that is less invasive than diagnostic surgery. So, Dr. Zernicki, I'll turn the presentation over to you.
Okay, thanks, Carol. Let me share my screen. And let's see. Go. So I'm gonna skip around a little bit, but I'm gonna talk a little bit and spend a little bit more time talking about what the immune system does in the normal breast. Because the immune response in the breast is really very active. And it's probably good for you to know what goes on in there. And then I'll try to link it back to how we're trying to use that to help generate that kind of an immune response to fight off breast cancers. But I ultimately think that we could take advantage of this immune response to actually prevent cancer. So let's just take a quick look at some of the players here. So, sorry about that. So there's a lot of different immune cells that are present in the breast, and they're present there at different times. So one of the players that has to be there is called the dendritic cell, or an antigen presenting cell. And it's kind of responsible for starting an immune response when you have a bacterial infection or a viral infection. It samples the environment and digests little proteins and peptides from whatever it ingests, and it shows it with a bunch of signals to T cells and B cells to generate your immune response. So that's one key player. Another big player in the breast is a cell called the natural killer cell. And there's a close relative of it called a natural killer T cell. That is a combination of a T cell and a natural killer cell. And there are these cells, this half of the figure here is what we call the innate immune response. That means these are the guys that come in very quickly and don't discriminate what kind of infection. They recognize different patterns rather than specific proteins or peptides or antigens. The other half of the immune response is what we call the adaptive immune response. That's what you're more familiar with, probably. The B cells that make antibodies and the T cells, of which there are two different main types, CD4 and CD8. And the CD8s are what are thought to be sort of a killer army. The CD4s are the master regulator. They help to regulate all this stuff and act as a go-between between the B cells and the dendritic cells over here. And the last player in the innate side are these group of guys called macrophages. And you could think of them as little Pac-Man. They gobble stuff up and they eat it. And they can also kill tumor cells. And you find them commonly in most cancers. They are in there as inflammatory cells. They can either suppress an immune response or they can activate an immune response. Lastly, I should mention that there are some bad actors or inhibitors that regulate all this, because you don't want your immune response to go on forever unchecked. You'd get autoimmunity. And so there are some cells that help to regulate this. One's called a myeloid-derived suppressor cell, or an MDSC right here. You see the red arrow showing it's inhibiting this. M2 macrophages and M1 macrophages are kind of opposite of each other. While they're not inhibitory, they mediate two different kinds of responses. The M1 is more associated with a virus response. The M2 is more inflammatory, inflammation, wound healing, allergies. On the other side, there's a CD4 cell that's called a regulatory cell that helps to shut down immune responses. And again, the MDSCs can inhibit a lot of these cells as well. So there's a yin and a yang going on in the breast all the time. If you were to get an infection, you would activate this part of the immune response. It would generate it would ingest the foreign invaders, and it would help to activate this half. When your immune response was fully active and controlled the invader, these inhibitory cells would help to shut everything down, to keep the immune response from just going unwanted. Now, there are multiple times in the life of the breast where this immune response plays an enormous role. And the one I want to just mention to you is a process called involution. So I'm sure a lot of you have had pregnancies or breastfed, and during that time you understand your breast undergoes tremendous growth, and new ducts are formed and new lobules, and milk is being made, and the breast is very proliferative. Well, when you finish using the breast, all that extra cell growth needs to die off. And in reality, that whole process is monitored by these immune cells, so that the immune response actually gets rid of any abnormal ducts and even some of the normal ducts that have been around that you were using during lactation or when you're breastfeeding. These guys here particularly play a key role, in this natural killer, what I'm going to call natural killer T cell, plays a tremendously important role in shutting down and causing the regression of all these ducts. Interestingly, I'll show you maybe a little bit later that this cell comes back around in breast cancer and plays a very positive role with getting rid of breast cancers as well. So this is the background. If you don't remember anything from today, you can remember that there's an innate immune response that does all the quick and dirty work up front. And there's an adaptive immune response that specifically helps to make you immune so that once you see it, you will be protected long term. And the same thing happens when you're given a COVID vaccination. These cells take up the COVID vaccine, they get activated, they then present COVID to these guys, and these guys then make you immune by making antibodies and having these T cells around. I'm going to mention one specific kind of breast cancer, and that is HER2 breast cancer. And as you can see in this slide, HER2 is a molecule that's on the surface through the membrane, and it signals cells to grow, spread, divide. Very active during breast duct growth, like when you're going through your teenage years and your breasts are developing, highly upregulated at that point. Also, HER2 and HER3 are highly upregulated when your breast ducts are growing during pregnancy. During lactation, there's another molecule called HER4 that's a little more expressed, and HER3 stays expressed. HER3 is a sister of HER2, and we call that the universal sister because it can pair with HER2, it can pair with HER1, it can pair with HER4, and mediate a lot of the effects of these guys. So in HER2 breast cancer, there's antibodies that have been made, such as trastuzumab and pertuzumab, or Herceptin and Perjeta, that are given to patients that have HER2 breast cancer. It binds to this HER2 molecule, prevents it from sending a signal. But the other thing these antibodies do is they also can help to activate part of the immune response, especially that innate immune response that I happened to mention to you, especially the cell called the natural killer and the natural killer T cell. So a lot of the effects mediating the signaling of trastuzumab and pertuzumab are through the immune response. Yes, it inhibits the signaling through HER2, but most of it is through inhibiting an immune response. And as a result, there have been many, many drugs and other antibodies that have been created all around the molecule HER2 and preventing a lot of its activity. People have now attached chemotherapy drugs to them. That's in the form of TDM1 here, and then the TDXD is another therapeutic antibody they're actually given to patients now with HER2 low breast cancer that has 5 chemo molecules attached to it. I'm going to show you something today that suggests maybe just the antibodies themselves can work if you use the rest of the immune response instead of throwing in all this chemotherapy. So right now, currently for HER2 breast cancer, we give those antibodies I mentioned, trastuzumab and pertuzumab. We also give chemotherapy because without the chemotherapy, the tumors don't go away. So it's been well known that you need a strong immune response in order for chemo to work. So we've thought that if that's the case, then we ought to be able to make the immune response work without requiring the chemotherapy. So we set about to take some of these actors that I'll go back to here. And the actors we're going to be talking about are the dendritic cell, the natural killer T cell, the B cell, and the CD4 and CD8 cell to get them active in treating breast cancer. So how do we do that? So we actually figured out that if we take those dendritic cells and we feed them little bits of HER2 protein, and then we inject them back into a mouse that has a HER2 expressing breast cancer, we can, to a degree, cause the tumor to slow its growth. And that's represented up here. You see there's a little change in the growth of these. But if we put them, instead of just giving them anywhere in the body like a vaccine under the skin, if we put them directly into the tumor and we add an antibody that's capable of binding to something on the tumor cell and activate these natural killer T cells, instead of the tumor just slowly growing, we're able to slow its growth, but then cause it to completely go away. And this was kind of a startling finding because it's the first time we were able to ever really make mouse tumors disappear. And we thought there was something special about giving the dendritic cells in the tumor that could. And if we didn't activate the dendritic cells properly, or we didn't give them the right antibodies, none of this would happen. So we asked the question, okay, we could make this go away. And by the way, if these tumors go away and we try to give that mouse that same tumor back, it rejects it. It won't grow. They're completely immune. So we did the same thing in an even bigger tumor, and the same thing happened. The tumor went completely away. Then we asked the question, well, what happens if we gave the mouse two different tumors, one on one side of the body and one on the other side of the body, and we did the same thing and injected them, but we only injected one tumor and not the other tumor. And in fact, this is the tumor that was treated. If you inject those cells I call the dendritic cell in the tumor, and you give an antibody with it, and the antibodies can actually even be Herceptin and Perjeta. In this case, it's a different antibody, but the tumor disappears. But not only does that disappear, at the same time, the tumor on the other side of the body that we're not treating goes away, which means that the immune response is getting everywhere in the body to eliminate anything. And that's an exciting thing for people that have breast cancer, because by treating one tumor, we can get other tumors. And in fact, we've done 3 and 4 tumors, and they all disappear equally. So we call this a smart circuit, because what we're doing here is we're putting dendritic cells that are activated. We take them out of the body. We can activate them so we can prime them so that they think the cancer is something like a virus now. We put it back in. Some of those cells stay there, but some of them move and they leave and they go to the lymph nodes, which is probably, in this case, the sentinel node. They'll drive a huge immune reaction in there. And then those cells now, because of the cells here, they pull other immune cells back into the tumor. They can pull in those NKTs and the B cells, and then the CD4 and the CD8 T cells. Not only that, some of them are going to migrate to other spots of cancer, and they'll help to pull these cells in with them to distant sites to get rid of all the tumors. So we actually took this into some patients and asked if somebody had a locally advanced HER2 breast cancer that was going to get standard chemotherapy with antibodies to trastuzumab and pertuzumab, we actually gave them some immune therapy with dendritic cells given inside the tumor with the antibodies, and the antibodies last for 3 weeks, so we only have to give them every 3 weeks. And we increased the doses of dendritic cells, and then we started with 50 million. We had done 20 million before, and now we're going to do 50 and 100 million. And then see what happens. We're going to biopsy and take an MRI, and then they would go on and receive their standard chemotherapy with the hope that if we got an immune response in there, that would make the chemo work better and make it more likely the tumor would be gone. Lo and behold, this is what happened when we did this. When you see this is an MRI of a breast, there's a very large breast tumor here. It measures about 8 centimeters. And these are positive nodes. It's in a different section than this, but these are positive nodes. They've been biopsied with a needle, so we know there's cancer in them. And then we gave shots in the tumor in the breast to the patient. You can see that even before we gave chemotherapy, the tumor's regressing dramatically, and the nodes are now all back to normal. So even though we gave the tumor here, remember with the mice with the 2 different spots, these nodes went away. And then after chemotherapy, you can't see anything left in the breast. This person went on to a complete response. Here's a couple more with a higher dose. This is about a 5 centimeter breast cancer. We treated it with just the immunotherapy, the dendritic cell shots, and the antibodies, and you can see there's a dramatic impact on this tumor even before chemotherapy. When she got chemotherapy, this went on to a complete response. This individual has about a 3 centimeter tumor. That one with just immunotherapy alone looks like it's gone. As you can see, the mass is no longer there, and you can see this inflammatory reaction in the background. So this was quite exciting to us. And in fact, we finished a phase 1 study, and every patient but one had a dramatic reaction to these injections in their breast and the antibodies, just like you're seeing here. We found out that the way the dendritic cells are activated may predict if somebody's going to go on and have a complete response. There was one molecule on them called CD141 that the higher that was, the more likely the patient would have a complete response. Those that did not, you can see the CD141 somewhat lower in those. And this just shows that there's 2 populations of dendritic cells. One, the blue one here, migrates. It's a smaller cell and it leaves and does its thing. The larger one here seems to stay in the area of the cancer and helps to pull a lot of the cells back. So this is kind of what a pathologist would see if you stain for different immune cell populations. And remember, I said there's T cells, there's CD4, CD8, B cells are CD20, the NK and NKTs are CD56. This is what a tumor looks like. It's very blue because it's all cancer. There's a few immune cells in there. After you inject them with DCs, you see this rapid infiltration of immune cells. And not only that, you see the dendritic cells filling up there. As you can see here, it's more blue than green. Here it's green and yellow, suggesting these CD141 and 11 positive cells in there. So in 3 weeks, you can dramatically change what a tumor looks like. So what does that mean for metastatic breast cancer? Well, we do know that a lot of patients, their tumors spread very early, and they go hang out in different places like the bone marrow. And even people with DCIS, we can detect disseminated cancer cells in their bone marrow even prior to them developing invasive breast cancer. So it suggests that breast cancer may have the capacity to walk around the body, and it may hang out there for years before it ever decides to grow to an active tumor again. And so we asked the question, what about these same injections we're doing? Can they help to control these cells to prevent people from developing metastatic disease? And lo and behold, we did just that. We actually took first from a mouse, we took those same dendritic cells and injected them into a mouse's mammary gland or their breast that has the capacity to develop breast cancer. And we could see that when we did that, there was an accumulation of these CD4s and a little bit of the CD8s. And then we could detect in their bone marrow that there's some cells that are not part of their bone marrow that express, for instance, HER2 and epithelial markers on them that we could say those are cancer cells. But if we vaccinated against them, we could make that number go down. We couldn't eliminate it, but we could make it go down. Equally important, though, is that we look to see what the cells were doing, and this is a marker of something called senescence. So when cells stop proliferating or they're paralyzed and they're never going to be allowed to grow anymore, they can become senescent. And this is what happens in the mammary gland when the process of involution, or the breast going back to its normal size after pregnancy, does. And it turns out that the immune response, mostly these CD4 guys and those natural killer T cells, secrete huge levels of a cytokine called gamma interferon, and they cause all these cells to become very blue because they're senescent. And if we get rid of the CD4 cells, we can reverse the process and make the cells back to the number they were and not be senescent anymore. So we took these cells that we get out of the bone marrow and asked, well, if we put them in another mouse, will they form a tumor? And the answer is emphatically yes. We can even take the numbers very low and they would become a tumor and they would metastasize. But if we took them from a mouse that we had vaccinated, they never, ever grew into a tumor. And we followed them out the life of the mouse, and they never formed a tumor. We even went as far as to say, well, okay, there's less cells there. Maybe it's just a number. We actually here gave the same number of cells as here, and they never, ever became a tumor, probably because they've been predestined to be paralyzed. So what that tells me is this immune response, through the production of a molecule called gamma interferon, can control the outcome of breast cancer cells. And this just depicts some of the organs. This is a liver, and we have lungs, and you can see these dark spots are actually what we call micrometastases formed, but not in the ones that we vaccinated. When we vaccinated them, we couldn't find any cancer cells in there. Only in the control mice that we transferred just untreated, disseminated cancer cells into those mice. So they do metastasize, and they do eventually grow up, and they will cause the mouse to die. What's more, we found a lot of the genes associated with metastasis were turned off because of the vaccinations. Things that were greatly turned on were now turned off, suggesting we could stop these metastatic cells. So that's an exciting thing. We've actually done the same thing in a human patient. I'm not showing you the data here, but the lines are exactly the same, where we've taken from a bone marrow aspiration at the time of surgery, found these disseminated cancer cells, and can implant them in a mouse with a no immune response, and they'll form a tumor just like that blue line right here. But if we expose the cells to interferon gamma, they never form a tumor. So we have the capacity to use the immune response early to try to eliminate metastatic breast cancer. That's a pretty exciting thing. Another thing you can do with the immune response is patients with breast cancer often take anti-estrogen therapy for years on end. And a lot of them, if they become metastatic, their estrogen receptor mutates so that it constitutively or all the time is turned on without having to even see estrogen anymore. And so they learn to how to beat the anti-estrogen drugs. And here is a group of known mutations that we can get from a simple genomic test that's commonly run by the medical oncologist that shows us all these different mutations in the estrogen receptor. And they differ by one single amino acid. They're all what we call a point mutation. And so we took all these point mutations and we asked the question, well, if they no longer exist in the body, would they look foreign to the immune response, and would the immune response recognize them, potentially go after them? And the answer is yes. So the green boxes indicate a positive immune response, and these are just different donors, healthy donors, that gave us blood that we could test. And these are all the different mutations. And you can see most of this chart is filled with green or yellow, and very few are filled with white. But in most cases, by taking these mutations, we could definitively show that the immune response could respond to every one of these mutations. We did the same thing in some breast cancer patients, and they responded very well. And we did the same thing in patients that had HER2 breast cancer, and they responded very well to these mutations. So much so that we then tested in a mouse, and what we were seeing, if we vaccinated against even the wild type, so the main estrogen receptor, just like we made proteins against HER2, we can make proteins against estrogen receptor, and they can help improve the survival of an estrogen cancer that's growing. Now, notice it's not going down in a way like I showed you with the HER2. That's because we're not adding any antibodies in here, because we don't have any antibodies yet to add to the ER positive, but we have some now that we're testing. And so some of the HER2 low antibodies can be used in combination with this. And there's a few others that we have that we're going to be tested in the lab soon. What about triple negative breast cancer? Triple negative breast cancer also susceptible to the immune response. Instead of the pro part of the immune response, people have created antibodies against the guys that are inhibitory. And one of them happens to be against this PD-1 molecule. And this is a triple negative breast cancer in a mouse that's growing just fine by itself. But if we add in what we call a checkpoint or anti-PD-1, you can see the tumor slows its growth. It doesn't necessarily eliminate it, but it does improve survival. The same also happens in patients with BRCA1 mutations, where they have a lot of mutations. They have a lot of immune response. The checkpoints can work very well in them. This is a group of patients that was heavily pretreated with triple negative breast cancer. So they had received many lines of chemotherapy. And yet, even with a checkpoint inhibitor, some of them had significant regression of their breast cancer. So this became an exciting end to triple negative breast cancer, in that we could now take immunotherapy and add it to chemotherapy in the form of this anti-PD-1, which is also known as pembrolizumab, combine it with chemotherapy as opposed to just chemotherapy alone. And what you can see is in patients who are treated before they have surgery, there was an increase in the complete response rate, which is associated with less risk of recurrence, up to about 65%. Our original chemotherapy numbers are about 30 to 33%. Another way to do this is to inject a virus that has a little immune stimulant in it. And one of our medical oncologists, Dr. Solomon, actually did that in triple negative breast cancer patients without any PD-1. He just gave standard routine chemotherapy. Interestingly, those that were either complete responses or had just a little bit of tumor left, they've never recurred. And for triple negative breast cancer going out into the 40 to 50 months, you're pretty much cured at that point. Unlike those that had a lot of tumor left, they still had some issues here. About 40% of them would recur. So this is another exciting area, again, pointing to the intratumoral delivery of something to manipulate the immune response. We've done the same thing with the dendritic cells. Dr. Costa put together a trial where we gave, instead of just HER2 on the dendritic cells, we gave them also HER3, which is higher on triple negative breast cancer. And we did that before chemotherapy. You can see here that if you give these tumors in some of the mice that have triple negative breast cancer, they can cause a tumor to completely regress. Others, not as good, although we're getting better with this one. But you can also see what it does in compared to chemotherapy or just the checkpoint therapy. The DC therapy is actually even a little bit better. So what Dr. Costa did is decide to give some priming dendritic cell shots in the tumor before people started their chemotherapy and checkpoint therapy. And this led to a very successful regression in the first patient that it was done for, and she couldn't even tolerate the chemotherapy, but her tumor actually shrank just with the shots alone directly into it. So we're continuing this trial. This is an exciting trial that's going on. We're now up to the 100 million dose, and we're hoping that we're going to see more complete responses than the 60% that we currently have. What other things can you do with this? You could try to do prevention. I mentioned to you that during the lactation and involution part of the breast, that's when breast cancers may get set up if all the bad cells don't disappear. Well, if we can stimulate the breast to get a strong immune response during that time, we might be able to eliminate all these bad acting cells and actually prevent breast cancer. And we're optimistic that we're going to be able to do that in something we call breast conditioning, where we can inject some activated immune cells into the breast, the dendritic cells, to drive these natural killer T cells to cause the senescence or elimination of any bad ducts that happen when somebody's finished finished breastfeeding. And this is just an example of how that happens. So I'm going to stop there because I'd rather let you ask any questions that you might have, and tell you that there is a day coming soon when we'll be able to just manipulate somebody's normal immune response to treat their breast cancer. And I think a day not too far in the future when we'll be able to use that immune response to actually prevent people from getting breast cancer. So thank you for attending this. I'd be happy to entertain any questions anybody has.
So Dr. Zernicki, thank you so much for a great presentation. A couple of questions that I had. At what stage do isolated tumor cells actually formulate?
So they can formulate it even in stage 0. We've actually got that, which is DCIS. We can actually pull some patients that have high risk DCIS. We can detect those cells that we call cancer initiating cells in the bone marrow of patients. That can happen anywhere in stage 0 to stage 3. Stage 4, they've actually become active metastasis at that time. So it can happen early and it can happen late. The ones that happen early, we have to act upon early. Usually there's enough time to stop them from doing something because they may not become active for several years. So if we intervene early, we may be able to basically stamp out the cells from becoming active metastasis.
That kind of falls right into the question that Karen posed. Can anything be done to DCIS prior to a surgery?
Yeah, we've actually done some trials, and we actually know of a high risk group. There's a molecular test we can run on a biopsy from DCIS to segregate people who have really what I would call high risk DCIS from those that have DCIS that may not even lead to cancer. And we've actually done some studies and injected those same cells in the body prior to surgery. And in about a third of the patients, their DCIS went away. We think with some of the newer things that we're doing and combining it with antibodies, we could make most of those DCISs completely go away and won't have to lead to a surgery. And hopefully that'll prevent any kind of further invasive breast cancer events or God forbid metastatic disease as well. So yeah, we're actively looking at that.
Thank you. And is this treatment specifically for breast cancer, or can other cancers be treated with this type of therapy?
So right now we're doing it for breast cancer, but in some of the lab animals we've done this with melanoma. It works very well. We've done it with other kinds of cancers, colon cancer. The same drivers that drive cancers in breast can drive cancers in other places like the esophagus, the stomach, the pancreas. And so eventually this kind of therapy can be applied to every cancer. It may require some other things added with it that may be specific for that particular organ, but we should be able to get this to work in any cancer.
Very good. Let me just make sure. And I believe that was it for our questions that came in. So we want to take the time to thank you, Dr. Zernicki, for taking your time and helping us understand more about what the immune system does when breast cancer is present. For those of you who have joined us, we are so excited that you have participated in this particular webinar. We do have a few more available for the month of October. You can go to our link that I have placed in the upper portion of the chat to sign up for more. And we look forward to having you join us next year. Thank you so much, and everyone have a great day.
Thank you.
Bye-bye.